For our next session, we have Professor Martin Donelly from Liverpool School of Tropical Medicine. And Martin has, again, we've collaborated for quite a long time, although he works on Anopheles and I don't even know what Anopheles is. But he's been co-leading the genomics of Anopheles efforts in malaria gen. So he's been involved in the genetics of the vector for a very long time. And without further ado, I invite him to give us his talk.
Thank you very much, Oliver. And yes, obviously, just to thank the GenRe organisers, and it's a great pleasure to be here. I've been a strong admirer of all the work which has come out of this network. I think it's been really some of the sort of the finest work we've had around the translational impacts of genomics. So it's very nice to be able to talk with you today. And I'm going to start my talk with an apology, because what I'll be talking about today is a lot of work on African malaria vectors. But really what I want to do is just tell you why that's the case, because if we look at the data which we've been able to generate on vector genomics, we have about 25,000 whole genome sequence from the two major malaria vectors, and unfortunately we only have about 300 from Anopheles minimus. We also have 300 from Anopheles dirus as well. So the resource availability for the Southeast Asian vectors is much, much lower, and that's why I'll focus my talk on the African situation. But really what I'd be very much interested in the conversation afterwards is do you think the technologies we're using in an African malaria vectors are relevant to the Southeast Asian system? Is there a priority list we should be drawing up about how many Southeast Asian vectors and which Southeast Asian vectors we should be looking at, what phenotypes should be looking at? I'll be very much focusing on insecticide resistance, but I'd be interested whether you had issues around behavioral phenotypes, outdoor biting we should maybe be addressing. So that's what we'll be talking about today, and I'll be talking about our genomics work in the context of two questions. And we've been using it to look at trying to understand what's the impact of malaria vector control on insecticide resistance evolution. And obviously the key question here is what's driving the emergence of resistance in this context? Is it the agricultural use of pesticides? Is it insecticides being sprayed on cotton? Is that selecting for resistance in the vectors? Or is it coming from indoor residual spraying, bed net use? Is that what's driving resistance? And obviously that's very important to try and disentangle, so we can start to think about resistance management strategies. If all the selection's coming from agriculture, we obviously have very different problems than if it's coming from indoor residual spraying, and we can think about rotations within our own context. And the other big thing we've been trying to address is trying to get a quantitative estimate of what's the impact of resistance on malaria vector control. As I'll show you, there's a number of observational studies which do suggest there's an impact of resistance, but it's very hard to work with national malaria control programs and give them a quantified estimate of this is what our resistance means and this is what we suggest you may wish to do in terms of interventions. And so just to start that conversation, so these are some data from Uganda. And it's just one of a number of observational studies which show how resistance may be impacting malaria vector control. And so work from Adrian Epstein and colleagues, and so these are various malaria reference centres throughout Uganda where we've been looking at malaria cases over a number of years and looking at the interventions which are going on in those communities. And really what we can see, and this is an area where the vector, Anopheles gambiae, shows extensive pyrethroid resistance and also apparently resistance to clothianidin. And what we're showing is that if you look across this time series, these are malaria cases plotted on this black line. The interventions are going in. Here we see a red bar where long-lasting nets went in, and we saw an increase in malaria cases immediately afterwards. So we didn't show any impact of a pyrethroid-based intervention on malaria cases. And similarly, we get over here in 2020-21, the long-lasting net distribution and spraying with a clothianidin-based product didn't seem to interrupt the seasonal increase in malaria. We sort of very successive numbers of malaria cases in this area in eastern Uganda in this year. So there's good observational evidence of the impacts of resistance, but what we're trying to do is think, can we move to a sort of more quantitative estimate? And we've tried to do that by doing contemporary assessment of malaria epidemiology and insecticide resistance. So this is a 5-country study coordinated by the World Health Organization, and it was conducted across these 5 countries. And so essentially what we're doing here is looking at a variety of surveys in human populations in various communities, looking for using infection cohorts, active case detection or prevalence surveys, doing large numbers of community surveys, and simultaneously looking at the impact of the presence of insecticide resistance using standard phenotypic assays. And so what we were hoping to look at here is, could we find the association? So we have some estimate of resistance going across in the mosquito population and some infection prevalence estimate or incidence estimate on the Y. What we were expecting to see in areas of high resistance, so low mortality in the vectors, we were expecting to see higher infection prevalence, and in areas with low resistance, higher mortality in the vectors, we'd expect to see this sort of negative correlation. So that was what we were expecting to see. And what we eventually saw, and these are data aggregated across these 5-country studies with some normalized estimates of infection prevalence, is we don't see any correlation. So each of these points is a measure of standardized measure of prevalence and an estimate of the resistance of the vectors in those communities. And we can see here there's no clear trend between resistance and infection prevalence. And so that made us sort of think that we were using a very, very noisy way of sort of characterizing resistance, and could we use genetics and genomics to make a sort of a scalable, much more sensitive estimate of resistance. And as I say, I'm going to be talking very much about resistance here, but I think we can use the same approaches for behavioral changes. We're starting to see sort of early biting in some of our vectors in Sub-Saharan Africa, in sort of early biting and sort of late morning biting. We could look at avoidance behaviors as well, but we're just taking insecticide resistance as our phenotype in the first instance because it's more tractable. And so what did the data show us? So these are some data from Anopheles gambiae. This is the 1,000 Genomes Project. What do we see in terms of resistance? And so, well, what we see is, so this is a mutation in the voltage-gated sodium channel. It's the target site of pyrethroids and DDT. And what the story is, that these pie charts are samples from across Sub-Saharan Africa, and the color is a resistance-associated variant. And so what we see is that we have some variants which occur and then spread quite widely throughout Sub-Saharan Africa. We see some which are constrained in some ways to East Africa. And then we sort of see these hotspots where we have multiple independent origins, parent origins of the resistance-associated variants. So this is the sort of story we get when we look at the genomic data. It's sort of relatively small number of resistance-associated variants. So in this case, there are 2 different amino acid substitutions, but multiple origins of that. And so that's the story about target site mutation resistance or knockdown resistance. And we see a similar story when we look at metabolic resistance. So you have 2 different types of resistance in vectors crudely. You have the mutations in the target site, and then you have these detoxifying enzymes which tend to show overexpression associated with resistance as well. And so we look at the genome sequence data. We can scan across the genome. So we have 2 autosomes in Anopheles and a sex chromosome. So what we're doing here is, this is an H1/2 statistic. We're screening across the genome, trying to identify areas with elevated evidence of positive selection. And really what you're seeing is that there's a relatively small number of loci which seem to be significantly associated with resistance. And if we dig down into them, what do we see? Well, what we see is that whilst there's a relatively small number of loci involved in resistance, there are multiple origins of that resistance. So it's not a story of which you might have in malaria of an occurrence of, say, PF-CRT76 and then universal spread throughout the P. falciparum population. It's multiple origins of the same or very similar mutations. So what we've done here is we've dug down into one of those selective sweeps, and underlying that sweep is a cluster of cytochrome P450s. And these are genes which are commonly associated with pyrethroid resistance. And what we say that so there's a cluster of these genes, and then we look in greater detail. What we find is that there's a massive number of copy number variations. So this is the gene, this wire and bead diagram is showing this gene cluster. And then we're looking at copy number variation across the genome. And what we're finding is that this particular region here is duplicated, some very, very long copy number variants, duplications within the genome, some much shorter. And what we're seeing is multiple origins. So the mosquito is evolving the same mutation multiple times, and there are about 38 independent origins of this resistance mutation in Anopheles gambiae. We know it's resistance associated. We know it's associated with increased expression of this particular gene. We know this gene has increased in frequency in populations from Kenya and DRC and Uganda over a sort of 3 to 5 year window. So we know it's showing a very marked change in frequency over time. And we know this pyrethroid through recombinant protein work can metabolize P450s. So lots of variation, relatively small number of loci involved, but multiple origins of that resistance. So what does this mean for Southeast Asian vectors? Is that story going to hold for Southeast Asian vectors? And I think the limited evidence we have suggests it will do. So let's take another example. This is the left arm of chromosome 2, and work from Sanjay Nadji in Anopheles gambiae identified a cluster of genes, these carboxyl esterases, which are strongly associated with organophosphate resistance. So this is a study conducted in West Africa, looking at telic spraying, IRS spraying. Thank you. In West Africa, and he found that these gene families were strongly associated with resistance. And then from a comparative genomic perspective, he just looked in Culex quinquefasciatus. So these two species, 150 million years difference in evolution. They split long time ago. And what we find there is exactly the same genes are associated with organophosphate resistance in Culex. So I think this gives us some idea that whilst there might be different origins of resistance, they're likely to involve the same numbers of same or similar genes. Getting slightly closer to home, these are data from Anopheles stevensii. I imagine you'll all be aware of the story of how Anopheles stevensii has recently, in the last 10 to 12 years, invaded Sub-Saharan Africa. It's originally found in Afghanistan, Pakistan, India, and Iran. But 10 to 12 years ago, invaded Djibouti, where it's associated with an increase in malarial cases in Djibouti. We've just started some whole genome sequencing work in Anopheles stevensii. These are some data from Afghanistan and Pakistan. And really what we're seeing is the same genes involved. So here's cytochrome P450. This is the one which had 38 copy number variations in Anopheles gambiae. This is the carboxyl esterase I've just spoken about, which is showing the same signals. And again, we're seeing the same patterns of selective sweeps associated with a relatively small number of loci. So slightly close to home, Anopheles stevensii also seems to have a relatively small number of resistance-associated loci. And even close to home, the only Southeast Asian data we have at present, this is work from Brandy St. Lawrence, and this is a FST-based analysis. It's slightly messy. These are data from Cambodia, but really what we're seeing here is a selective sweep around a cluster of glutathione S transferases, which again, commonly associated with pyrethroid resistance and DDT resistance. So I think there's a lot of prospects that if we take the same technologies we'd be using in Africa and apply them to Southeast Asian vectors, we're likely to be able to use to develop powerful resistance-associated variants in Southeast Asia. And as I say, that's be interested to talk about what species do you think should be on our priority list. As I say, we have some Anopheles dirus, and we're going to be refining some of the work around Anopheles minimus coming up, but we're very interested in hearing and collaborating on new variants, new species. So we now have a reasonable understanding of what's driving resistance in Anopheles gambiae. What we're interested is now moving that genomic data into sort of more a translational domain. How do we use it to inform control policies? How do we address the issues of what's driving resistance and what's the impact of resistance on malaria epidemiology? And we're doing that in a number of studies by embedding genomic screening into vector control programs. And this sort of cartoon shows what we're trying to do. And I'll talk about one specific trial. But essentially what we're doing is simultaneously collecting mosquitoes embedded within randomized control trials. We're genotyping or whole genome sequencing those mosquitoes and characterizing their resistance patterns based on their genotypes. And then we integrate those genomic data into the standard epidemiological models to see whether they improve the fit. Does understanding resistance on a cluster by cluster basis across a trial result in improved fits, and can we then quantify the impacts of resistance on epidemiological outcomes? And so to talk about the sort of first study where we've done this in a trial in Uganda, and this is called the lineup trial. It was a trial of two different types of bed nets, or two classes of bed nets, your standard pyrethroid-only net and your net which had a pyrethroid and PBO. And PBO is that compound which synergizes and can knock out the impacts of some of the P450 mediated mechanisms. So it's sort of the next generation of net, which should have some resistance management properties. So it was about 16 million nets were distributed across approximately 40% of the surface area of Uganda. Our primary outcome was parasite prevalence in children less than 10 years old. But we had a variety of secondary outcomes, including vector density, and I'll talk about this sort of secondary outcome in greater detail because we've done some new analyses on that very recently. So two-arm trial in essence, and what were the results? Well, the results were, if we look at the primary outcome, in those communities which received a PBO-LNIN, one of these second-generation insecticide-treated nets, there was about a 20% reduction in parasite prevalence in children under 10 in those communities. So good evidence of impact of next generation nets, and an even more marked impact on vector populations. So in the same households we are following of the cohorts of children, we're also doing mosquito density collections. So what we're showing here is that there's about a 73% reduction in vector density in those same communities. So good impact of the interventions, or the PBO interventions on mosquito densities. So we've got good evidence of impact. So the first question we wanted to ask is, do we have any evidence of resistance marker changes over time? And so what we did is just a candidate marker screen. So each of these is a time series across the trial. So we have baseline, and then we did surveys at 6, 12, 18, and 25 months. And we're looking at marker frequency here. So these are just a series of resistance-associated variants. And really what we're seeing, the ones in green are ones which showed significant increases over time. So over the course of the 25 months of the interventions, we saw significant changes in resistance marker frequency. And because of the range of communities we're looking at, I think we've got good evidence this is driven by the intervention. So we do show that the interventions are selecting for resistance. It's not just agricultural use or other use. What we also saw is some replacement. So this marker in red is showing to be decreasing in frequency, and that's because this marker is increasing in frequency. They're on the same codon within the target site, and we're seeing this behavior. The data were also really very useful for screening changes over time and understanding the distribution. So this is a schematic of Uganda. We're looking at one particular mutation, and really what you can see, this is the frequency change over time, and what we're seeing is this sort of leaching or bleeding in effect from neighboring DRC. And so over the course of 25 months, we see a resistance in this area increase from around sort of 15% to around 45%. So we've got good evidence of origins and spread, so we can start to think about how do we interrupt the spread of this variant using these data. So I think this sort of embedding genomics in trials is a good way of looking at sort of the impacts of the interventions on resistance. But we've also started to think of the opposite. How do we look at the impacts of resistance on the interventions? And we've been using an approach to generate genomically estimates of resistance using this sort of polygenic risk score-based approach. So essentially what that does is it's very similar to the traffic light system that the GenRe are using this approach of trying to quantify a mosquito. An individual might have a variety of different resistance mutations. How can we put that into a quantitative concept? So a mosquito population which has a high frequency of all the variants would be sort of red on your GenRe map, and one which would sort of have a few might be green on your map, if you will. And so it's just quantifying that, really. So we get this estimate of resistance, a polygenic resistance across our populations. And you can see here, this is sort of a standardized scale. But we also have the estimates of household density of Anopheles gambiae. So what we really want to try and see is, does evidence of resistance at baseline impact our evidence or our mosquito densities? And the answer is it does, really significantly. So what we've done is just looked at that secondary outcome. We've looked at Anopheles density. We've rerun the epidemiological models. And essentially, we've just put in this estimate of genomic resistance. And really what we find, again, still evidence of significant impact of the PBO nets, significantly associated, in this case, 65% reduction in Anopheles density, as we did. So we sort of regenerated the initial score. But what we see here is that this polygenic risk score is associated with a 24-fold increase in mean Anopheles density. So it's clearly evidence that the higher polygenic risk score at baseline is associated with higher density. So we're starting to show this impact of resistance. So we now have a sort of a framework for thinking about how resistance impacts. And much like you've heard in the talks this morning, we do now have panels of resistance variants for Anopheles gambiae, and again, I think these could be very readily developed for Southeast Asian vectors. So we have the vampire panel, which is essentially a collection of resistance-associated variants, and associated with that, we also have this AMP-seeker pipeline, which actually takes the outputs from your Amplicon MySeq panel, processes them automatically, gives you sample maps, standard PCAs, allele frequencies, and ultimately it'll output this polygenic risk score approach. This is specimen agnostic, so all we need to do is slot in a new reference genome. So if we wanted to use this for Anopheles minimus, Anopheles epiroticus, all we need to do is add in the reference genome, and you'd have the same facility to do this in other other vector species. So to conclude, I think we've got good evidence of, by embedding genomic surveillance into randomized control trials, we've got a powerful way of addressing multiple hypotheses about the impacts of malaria control on insecticide resistance. And while the data are tentative, and we're still working through both the malaria incident prevalence, from the vector density data, we have a framework for understanding how resistance is impacting intervention. So we can start to make evidence-based advice to national malaria control programs about what resistance means and how that should impact the interventions they're selecting. So just to conclude, what does this mean from a sort of a Southeast Asian perspective? I think there's a really high likelihood of the approaches taken in Anopheles gambiae and Anopheles fenestus and Anopheles stevensii will work very readily in Southeast Asian species. Resource development is underway. As I say, we have some minimus and dirus sequences, so hopefully within 6 months we're starting to sort of have the resistance panels for at least these two species. And we do have these sort of species agnostic analytical frameworks, the AMP-seeker pipeline, which can be very readily translated into Southeast Asian vectors. So these are the sort of the positives. Obviously, I'm hesitant to tell you this because you know far more about this than I, but obviously you've got much more diverse vectorial systems here. As we were talking about this morning, many more diverse, we're very much working in a falciparum-only habitat. You've got challengingly and interestingly, sort of much more diverse transmission ecologies. We're very much working in rural environments with our vector species, but you obviously have this work-related exposure, forest fringe malaria, which is something we don't deal with. And obviously you have much more diverse vector biomics. We have working with species which are essentially pretty much anthropophilic. We don't have sort of the same challenges you face here. But I think the approaches we're taking using resistance as a phenotype would be very relevant to addressing sort of some of the issues you have here. So I think my time is up. So really just remains to sort of thank you for your attention, thank you the organisers for the invite, and very happy to take questions. So thank you.
Thank you, Martin, for that was a very clear and comprehensive perspective. Before I open to the questions, can I just clarify something? So when you say you just slot in another reference genome and the pipeline is agnostic, we're just talking about the sort of genotyping analytical part. The primers and all this will have to be specially designed for whatever species we're looking for, correct?
Yes. Yeah.
Okay.
So yes. So the assay development will and you also need to specify which loci you're interested in it. But yes. But once you've got the reference genome and your reference candidates list, you can just port it, and it doesn't need to be Anopheles. You can use it for falciparum. You can use it for
and realistically, how long do you think that discovery phase might take here in Southeast Asia for some of the species we're interested in?
We are looking at, so we're hopeful within 6 months we'll have the first. So the minimus data are already been published, but they haven't been phased. So the first thing to do is phase the minimus data and then do the H1/2 analyses, and we'll hopefully do that early next year. The direct specimens we're going to put a postdoc onto, and so hopefully within 6 months. So I'd hope by the second or third quarter of next year, we would be able to sort of share with the community some sort of candidate loci. Those specimens aren't phenotyped, so we wouldn't have the genotype-phenotype association, but I think they're likely to be the same loci, and I think sort of they'd be very, very strong candidates. So.
Great. Sorry to have barged in like that. Questions for Martin?
So,
Luke one?
yes. Also,
Yeah.
Okay, so many thanks for sharing this very informative presentation. And in fact, I always have some questions for my colleagues from Africa, from Ifakara Health Institute, such as Prof and GenRe-Mekong from Liverpool. So, for my understanding, for the malaria vector, we have done a lot of efforts, particularly in our project. We also put a lot of efforts and budget for malaria vector surveillance. But I always have several questions going for them. So normally we try to use the malaria vector data to understand the transmission hotspot or to understand the intervention impact. So my first question is, how to use the vector data to optimize our intervention? Should we have different interventions for different target population based on the vector surveillance data? So the second is about, is it possible we can use the vector data, including the genetic data, through the modeling to identify or to predict the hotspot? As far as when we work even in Africa, although the prevalence is high, more than 20%, but there is still some hotspots, some specific villages with high transmission, although a lot of interventions. Better net coverage is high, and better net use is high, but the transmission at that particular area, village, is always very high. So we are not clear. And the third is about the behavior change. As long outdoor biting, outdoor transmission is a big challenge, even in Africa. From my experience in our project area, almost 80% transmission from the outdoor bite. We try to use model to understand. So is there any relation between the insecticide resistance and the behavior resistance? So because in China we have different experience, we put a less budget or activity for the vector surveillance. We put more efforts and budget going for the interventions, not much budget for the surveillance, particularly with the limited budget, limited resources. So this is, I always conflict with my colleague from Africa. So so far I cannot understand these questions very clear. Thank you very much. Yeah.
Right. So maybe take your second question first. Can we use sort of genomics and other surveillance data to identify hotspots and understand the hotspots? I think we can, but we're only just starting to think about the appropriate sampling frameworks to identify hotspots. So we work with colleagues in the University of Lancaster to design ecologically explicit sampling frameworks. I think a lot of vector surveillance is opportunistic. We go and visit the same villages each year. We have sentinel sites. They may not be representative of the environment as a whole. So, for example, in Ghana and Kenya, what we've been doing is using ecologically explicit sampling frameworks. We gather sort of data from satellite imagery, we identify the habitats, and then we overlay our sampling framework. And that is starting to show marked differences in vector species distribution, marked differences in resistance patterns in relatively small spatial scales. So I think we can be moving towards those sort of focal control. The issue is whether the National Malaria Control Program has the ability to do sort of targeted control, or is it very much reliant on universal coverage campaigns. So I think that's a sort of an issue we haven't really dealt with. But I think the technology is moving towards hotspot identification. And then using adaptive sampling frameworks, that you maybe use that framework for sampling so many villages in the first year, and then you keep some the same, but you move some, and then you can start to delimit the hotspots, and that's what we're working towards, but it's still some way off. Whether there's an association, question 3, whether there's an association between insecticide resistance and behavioral resistance, we really don't know. What we're starting to show is that what we think are behavioral resistance variants are changing in frequency over the course of the interventions. So, for example, in Anopheles stevensii, Gambi, and Phonestus, we're starting to see a gene which we think is associated with behavior. It's a DGK kinase, which is changing in frequency over the course of an intervention. So we're starting to see other variants coming through which might be driving those behavioral changes. The problems we have with quantifying those behavioral changes is we don't know they're fixed in the we don't know they're fixed heritable phenotypes. They could just be plastic. So it's a very, very tricky system, but we don't have the evidence as such. And your first question, Tanzania, what was the first?
But in fact, for the intervention, we use the same intervention. We don't change some intervention. Also, we have a lot of work the species and work the distribution and work density. But in practice, for the intervention, it's same.
Yes, I don't think we're there yet. I think the case there is probably Anopheles stevensii invading into the Horn of Africa, which used to be an Anopheles Gambi and Phonestus dominated habitat. Anopheles stevensii is spreading, so it's clearly the interventions we're using against Gambi and Phonestus aren't working. So I think we're starting to get there, thinking about sort of what additional interventions. So specifically with Anopheles stevensii, it has a slightly different ecology. It breeds in big water storage containers, so we're starting to think about the use of larviciding. So I don't think it's going to be sort of one intervention in different places. I think it's going to be layering on interventions in specifically where we see an additional vector species. So we'll probably still have universal bednet coverage campaigns, but in sections of Sudan, we might have larviciding based approaches as well. So I think it's going to be an additive process, but yeah. Okay,
so I'd like to interrogate some of the other control programs. I think the Kwangs here have been wanting to ask some questions.
Yes, just one question.
It's on.
Thank you very much for your presentation. Very interesting, informative, and very useful for us. I don't know how long you conducted your studies in Africa, how many years, but I saw that the very big sample size you collect, maybe more than 80,000 mosquito you collected and do molecular techniques. But in Vietnam, so we have a limited budget. We cannot do the sample size like you like that. So I will wonder to ask you about your experience how to collect the mosquito to do the PCR or sequencing technique, because you know many mosquito outside. We can collect more than thousand mosquito, but we cannot did the PCR technique or sequencing technique for all the mosquito we collected. It's the first question. And the last question is about the transmitted in the forest. I am thinking we not use insecticide in the forest. And every country in the GMS region in this morning says that the highest risk factors is the people go to the forest and get malaria in the forest. So I think what is the residual resource in forest, animal or people or Anopheles? I think Anopheles cannot because their longevity is very short, only under 1 month. So what is the receiver of malaria parasite in the forest? In order to, whenever people go to the forest and get malaria immediately. And that's my 2 questions. Thank you.
I'm not sure I can answer the second one. Certainly in Central Africa, there are potentially primate reservoirs of some of the malaria infections we see. So I don't know if that's, I'm not going to put my neck out on the line and say that's the situation in sort of, aside from the primate malaria. So I don't really understand and that's really my ignorance, I don't really understand the forest transmission cycle at all. But it's, yeah, very intriguing how you get sort of persistent transmission in these settings. In terms of the question around sort of trapping collections, and particularly trapping outdoor vectors, we have the same problems with, certainly Anopheles stevensii in Sub-Saharan Africa. We've done a range of Procopac-based aspiration collections of vegetation, of household structures, of sort of cattle resting structures, BG Pro traps. We're doing a lot of stuff around trying to understand which are the most effective collection techniques. We're not there yet, I don't think, and I think it's sort of a location by location setting. So even when we compare Ethiopia and Sudan, some of them Djibouti, some of the collection techniques just don't seem to yield the same number of vector species as we move from essentially contiguous countries. So it's a real problem for us as well. So understanding what's the appropriate sampling is something we're very happy to talk about and sort of share our experience of what has worked and what hasn't. But again, I've given you 2 no answers there, I'm afraid. Sorry, but.
And you have any information about KDR chain detected in Vietnam or any in the GMS?
So many years ago, as I say, I worked with NIMPI up in Hanoi, and we did some Anopheles epiroticus sequencing. And so we've got some whole genome sequence data from Anopheles epiroticus, and we do have KDR in epiroticus, which Lian Tran did many years ago, but we haven't done anything more recently than that. But very happy to share those data. And so those are the specimens which contributed to the 16 Genomes project. So those data are freely available, but no one's investigated such.
Thank you. I wanted to ask. Sorry. Oh yes. Yes.
Yeah, I had. Thank you for the very comprehensive presentation. But I have some question for you. Is Southeast Asia, especially for Vietnamese, for Vietnam, you have the isomorphic sibling species. It related to behavior of writing, behavior of breathing blood, as well as vector capacity. So, do you have information about the whole genome sequencing or something about to relate with it. For the resistance of insecticide, we know that many kind of the insecticide chemical mechanism. But as you know, I think there are something similar with parasite, malaria parasite. We don't have changed the insecticide for maybe more than 25 years. Now we still use permethrin. Yeah. So what is different between the way we treat it with the vector when we use already resistant and not yet resistant? It's not so different between them, because I think still cover of the population or something like that, so no different. And up to now we don't have any chemical to replace the permethrin or something like that. So what do you find when you use with resistant sequencing or something like that? What is the main purpose for it?
Sorry, so I understand fully. Your questions around the isomorphic species you have in Vietnam, which have different resistance associated.
Yes, I think so.
And the second question was around, you've had 25 years of pyrethroid use, and you have resistance in your mosquito populations, or you?
I think the treated with the population is the same between the already resistant and not yet resistant.
Okay, so okay, yes. So with your sort of isomorphic populations, I think these approaches would very rapidly pull those apart. So, example, the resistance panel we have, which is for Anopheles gambiae, there are 5 SNPs on that panel, which will tell us which of the members of the cryptic species complex are there, and I think that'd be very readily able to do. So have some resistance markers which are sort of barcoding, if you will, which pulls apart the species. And I think that's easy enough to do. In terms of how you might wish to think about deploying different strategies for different resistance populations, I think it's now worth thinking about both the PBO nets. So the trial I spoke about in Uganda, that was the first of 3 trials. And what we've done is we've compared standard nets to PBO nets and shown that they had an improvement. We've then compared PBO nets to pyriproxyfen nets, and they're just about the same. And now we're in the process of comparing PBO nets to chlorphenapir nets. And so now we do actually have an understanding of, in very resistant populations in Uganda, which works best. So I think you would be able to work with the NMCP to actually say, okay, this is a highly resistant populations. We would advise using chlorphenapir-based nets in that situation, because they do have a marked impact in resistant populations. So I think we can start to think about structuring that. It may be worth thinking about, and that's what the National Malaria Control Program in Uganda did, is they gave the nets out in a randomized fashion. So they actually worked with us to do a randomized distribution. So you could actually maybe do within Vietnam, if you have more than one net type, to think about building a trial into the design. So you could actually get tailored evidence of how the different interventions are impacting your different populations.
Thank you.
Great. You know, what are your questions with respect to the Anopheles genetics? And I mean, this is what has been presented here of interest to the work that you are doing?
For the vector control, very important and useful for the malaria control program, and like nearly eliminate, right? But in my country, for the entomology section in Simpe, we concern like insecticide resistance, because we provide the long-lasting net to prevention, yes. But we think about the future, if insecticide resistance, how to change, how how to sort the another chemical. We think about this the first, and second, and for the genetic Anopheles, we don't have the information. We don't have the activity. We only collect the mosquito to analyze the species. Like in my country, we have the Anopheles minimus maculatus, but another species, but not too much. But really important for us. Yeah. Yes. For this.
Thank you. Dr. Siv, can I get you to comment on this? You've been unusually quiet on this.
No, I had two things. One, just echo my question again to you already, that does the Asia or Cambodia's Anopheles have capacity to carry the African parasite to transmit Cambodian people? And because I have a question that doubt that some species has been a primary vector in this country, and sometimes it becomes the same species become secondary to other country. The same as for Malaysia and Cambodia. Malaysia, Anopheles maculatus is a main malaria vector, but in our country, maculatus still a second. It's not very primary like Dirus and Minimus. So because of what? Because of the parasite different in different location, or due to what? So it's something like this. So that's why now we have still concern that does our vectors have ability to transmit some parasite. And the thing is talking about genomic vector here. For Southeast Asia, we have been known that we are forest malaria. We can say that's forest malaria because most of our vector is linked to forest. And now in our Mekong country, malaria case has been dropped down dramatically, and transmission now is happen in the forest. It means that they go to the forest. Their transmission is not really in the village. So the role of vector control in the Southeast Asia for long lasting seem to be that not very clear, because the long lasting we use in the village, but not really in the forest, because people visit to the forest. Sometimes they work in the night time, no time to sleep in the under the bed net or something like that. So we use another hammock net to change their lifestyle during that. So for me, it's insecticide resistance has been noted in the South Asia since before 5 or 7 years ago. We have a trouble, but now when malaria case is become lower, resistance seems to be that reluctant, because we are not mainly bed net is not really work on the malaria elimination, because we need to tackle people go to the forest. How can you protect these people by providing chemoprophylaxis and something like that. So for me, I just want to know that how can we apply the genomic entomology from Africa to Southeast Asia, because seem to be complex and also different. And the third question is continue with the one question from Vietnam about the hotspot. Currently, in South Asia, in order to identify hotspot, we need to identify the source transmission. And does this hotspot really have a source transmission? And you have been told that entomology is a collection mosquito. Sometimes it raining, you cannot collect, or sometimes you cannot collect mosquito, can you say that there is no anomaly? Or no, because very very complicated until you are do several collection routinely, so you can know that these areas have a main vector or don't have or something like that. So in our study right now, when we identify want to identify a short transmission, we just collect mostly only 3 days. So if they're present or not present, we ask say oh, they're no present, there is present or something. So we use some score for how to intervention on that. So this is a quite challenge. So how vector genomic surveillance can predict hotspot? I don't know modeling how they're doing, because some people they are overlay satellite, they overlay breeding site, and also source of breeding, and they say that the breeding is these areas look like Anopheles diverse breeding site. So when they overlay that, they just say that the is possibly have main malaria virus. Can be Genomic also can do that for that? Thank you.
So yes, I think the habitat suitability approach of using sort of remote sense data and predicting habitat is very, very crude. And I think if you're looking to identify hotspots, it's probably looking at genomics in the plasmodium to look at transmission networks, which is probably the more powerful approach, particularly if you're finding problems actually sampling vectors in the area. Obviously, we can't do the genomics if we can't find the mosquitoes. So it may well be that it's not a useful tool for looking at sort of outbreak response. But if you have sort of more consistent hotspots of transmission, I think then, yes, regular sampling of genomics and using this adaptive sampling approach can, and that's what we're trying to do, we've done in Côte d'Ivoire and Burkina Faso, you can actually start to delimit the hotspots. So you can actually say, okay, that you're consistently finding vectors here, you can characterize them for their resistance genotypes, and then start to think about targeted control. So I think there is a place for it there, but probably not in outbreak response where you just have to select for 3 days. Your question about can Cambodian vectors transmit African parasites? There has been some work by, so Brandy St. Laurent did some work about looking at sort of transmission and showed that there was, for at least for falciparum. So I'd be sort of confident is perhaps the wrong term, but I think it's likely that Southeast Asian vectors can transmit African P. falciparum. And there's quite a large number of studies about using Anopheles stevens and other vector species with a variety of parasites for falciparum. Probably Vivax might be more sort of tricky, given we know there's sort of transmission, South American and sort of South Asian transmission groups, which are less readily permissive in Anopheles species. I think that's another question, but I think falciparum is likely to be, but I can't swear to that. But yeah, very interesting question I think we need to address. And your point about Anopheles maculatus and why it's a secondary vector in some areas and primary in the other, I guess that goes to we need to go out and sample and look at the genomics of it, because I think just learning from the Anopheles gambiae, when we first started, we thought we had 6 species in the Anopheles gambiae complex. We are now up to about 9 or 10, just because we're starting to throw up new, very localized populations in coastal Tanzania, in coastal Gambia, in some places in Central Africa. So I think it may well be the maculatus is like your colleagues in Vietnam were saying, a sort of a series of isomorphic species, and I think we just need to sample and find out, really.
Am I right to say that, in fact, it was Brandy herself who said that once you started looking at what species were there in Cambodia, all that was known beforehand didn't really hold up. You know, it was, the balance really wasn't what was expected,
yeah.
Right?
Yeah.
So there's probably a lot that isn't properly sampled yet.
Just one more question: that do you have been looking for Anopheles vagus in our Southeast Asia here? Anopheles vagus here, you look at the microscope, you cannot tell that this is Anopheles vagus or Anopheles gambia. When you go to the genome, and only one of the genome different.
Yeah.
Yeah. So for me, Anopheles burger is can breed everywhere, in the right field, even Cambodia.
Yeah.
Yeah. So but now it's still not a primary vector. But there is a Gambia primary vector in Africa. So I'm so I don't know. Do I've been looking for that?
Short answer is no. That's very much news to me, but we're very interested in. I think it's one of the questions. Looking at the priority list of what Southeast Asian vector species we should be looking at, and that sounds like a clear question to address. A frightening one, but
So we still haven't heard anything from Thailand, from the VBD. I don't even know, are the species on the Thai-Burmese border the same as on the other side? Hey, use the microphone. Sorry. Yeah.
I think maybe difference. Yeah. However, no problem. I have one question about. I want to ask you some of your experience, from your idea about the. Do you have to test the bioassay test or the susceptibility test, and you compare with the molecular mutation in the mosquito or not? Like, do you have the experience about that? The result from the bioassay test is related with the molecular mutation or not, something like that, when you found the resistance.
yes. So we do initially. So we've done a number of genome-wide association studies whereby we take resistance phenotype mosquitoes, and what we try and do is take the most susceptible, so they might be killed by a 5-minute exposure in a WHO test tube, and then we take the most resistance, which might survive 2 hours in a WHO tube test. And then we do genomics on both of those populations and try and find resistance-associated variants that way. So yeah, so these markers which show the big selective sweeps, we do have resistance genotype phenotype association with them. But I think because they're so common across different species, we can probably move forward without doing that intermediate, that sort of genotype phenotype approach, assessment approach, really, because I think there's such strong evidence from other species. The fact that Gambi and Culex quincunfasciatus have evolved the same resistance to organophosphates, I think if we found that in Anopheles maculatus, yes, we could do the genotype phenotype approach, but I think it's probably not necessary as such. We can move much more rapidly to use it. And I think the problem with the phenotyping approach, it's very, very noisy. It's subject to variations in temperature, humidity. We had one instance where one of the technicians used to go out for a cigarette and then he'd blow the mosquitoes through. And so I think they all died of nicotine exposure rather than pyrethroid. So there's all sorts of environmental variation in those tests, which are very, very noisy, which prevents us doing that accurate genotype phenotype approach, I think.
Thank you. And other... Oh, it's that one. Dr. Wang still has one. Okay. You know, Marty's never going to come back again, you know.
Thank you so much. Very informative presentation from the Professor Martin. Regarding your publication about have a study cooperation between the Liverpool School Tropical Medicine and NIMPE and at that time, so far I remember more than 15 years ago. Yes, and only one publication. And at that time, your school do together with us in the Central Highland of Vietnam. But I think up to now, after 15 years ago, under weather and under global climate change and under the insecticide pressure, and therefore the population of the mosquito change step by step. And up to now in Vietnam, we have two provinces have an outbreak of the malaria. but very stranger for the this outbreak, and therefore I think you can come and you can find out in Vietnam, especially in the appear or occurring of the blood-borne malaria is the new species for parasite, and any other problem between changing from main vector to secondary vector and from secondary vector to the main vector or not looks similar to the Malaysia situation 5 years ago.
Okay.
Yeah, I warmly welcome you to Vietnam.
That's lovely to hear. That's the reason I'm here, actually. I remember my time in Vietnam very fondly, and that was obviously through a British Council link, which didn't have much money for research, but I'm very keen to restart collaborations in Southeast Asia. So thank you for your invite, and hopefully we'll find a way of finding the funding to sort it out.